This is a lecture about basics of EEG, and I will present some global mechanisms that are essential in the generation of these rhythms, because that is what we typically observe if we record an electroencephalogram. Let's start at the elementary components of our brain, which are the neurons. And we've extensively discussed this in the previous lectures. And shown here is a layer 5 pyramidal neuron. And let's assume that we perform some measurements indicated here with these electrodes, which we can perform at the dendrite or at the soma or in the axons. And we learned thus far that this will allow us to record action potentials if the neuron is sufficiently active. And we learned a lot about how those action potentials are generated. And here in this illustration, this is recapitulated. The axon is the action potential, as for instance recorded in the soma or the axon or in the dendrite. And what is nice here is that you can see that the shape of the action potentials is slightly different in the different parts of the neuron. However, this is not very essential for our current discussion, but it is good to know that there are some differences in how the action potentials look like depending on where in the neuron they are generated. But in principle, you can measure them basically in any part of the neuron if that part of the neuron is sufficiently activated by a synaptic current. And we also learned that we can describe action potentials using the Hodgkin-Huxley equation, shown here on the right, where the I here, the current, can be an external current, but in the central nervous system, this current is typically realized by the chemical synapses. Shown here again on the slide, the chemical synapse that excretes neurotransmitters in response to an action potential, and not shown here, but it may arrive here at the presynaptic part, resulting in a release of neurotransmitter in the vesicles shown here into the synaptic cleft, and that can interact with the postsynaptic receptors, the ligand-gated receptors. And we also learned that we can describe the change in the conductance induced by the synapse, can be described, for instance, with the alpha function shown here, which is, of course, then related to the alpha synapse. And indeed, we learned that if we record in single neurons, we cannot only measure action potentials, we can also measure rhythms, trains of action potentials. So indeed, single isolated neurons can generate rhythms, which are more or less periodic fluctuations in the membrane potential. And these rhythms can be generated depending on the particular kind of the neuron, if the neuron is stimulated with a sufficient current, but it's also possible that neurons generate rhythms spontaneously without an external current. Here in this case, the rhythms are essentially the regular appearance of spikes, which is a train of action potentials. At the same time, the neurons may also show membrane voltage fluctuations in a low frequency range. It's to some extent illustrated here. This is a membrane fluctuation, and these are the spikes, and this is much slower. And in principle, it's possible if you stimulate the neuron below threshold that you will only measure membrane fluctuations without the occurrence of spikes. So it's good to realize that two kind of rhythms in that sense can be recorded: the spikes and the membrane fluctuations without the spikes. We also learned that we can reduce the four-dimensional Hodgkin-Huxley equations to a lower dimension. This is an example of the reduced Hodgkin-Huxley equations. That still captures the essence of an excitable cell, of an excitable neuron, that is the generation of action potentials. And the advantage of this approach is that it makes it possible to do the analysis in two dimensions and to do the analysis in a plane, which is relatively easier than working in 4 dimensions. And this is an illustration of how this reduction of the Hodgkin-Huxley equations indeed can still generate trains of action potentials. It's slightly misformed. The downslope is a little bit slower and the width of the action potential is a little bit larger, but the shape pretty well resembles the shape of the action potential if we would have used the 4-dimensional system. And we also learned that if we use electrical recordings with an electrode placed inside the neuron, that we can not only measure these action potentials, but also the slower changes in membrane potential, subthreshold changes in membrane voltage that are known as excitatory postsynaptic potentials or inhibitory postsynaptic potentials, depending on the kind of the neurotransmitter that is excreted, and essentially the nerve potentials associated with a particular ligand-gated channel. So the current in the Hodgkin-Huxley equations is typically realized by the chemical synapses. And what I will show you now is a recording of this electrical activity, not exactly measured inside the neuron, but a little bit placed outside neurons. And those recordings are known as the measurement of local field potentials. And in this grid, in each rectangle, the recording of a particular electrode is shown, and this is actually a recording from a monkey in the cortex, used to explore particular patterns in these local field potentials measured over a particular area during performance of particular movements, motor movements of the monkey. And this is how it sounds like. This is a zoom in. And this is the average of multiple local field potentials. And it very much looks like an action potential because it's recorded very close to the neuron, but it's not an intracellular recording, it's an extracellular recording. So we discussed shortly the measurement at microscopic scales by placing an electrode inside the neuron that allows us to measure spikes, action potentials. And this is a postsynaptic current, for instance, induced by the excretion of GABA, which is much slower. So that is basically subthreshold membrane fluctuations, and this can result in the generation of spikes. The movie I just showed was a recording of a local field potential where an electrode is placed very close to several neurons, and that allows us to record spikes as well, but not necessarily from a single neuron, but from multiple neurons, and essentially reflecting the various action potentials that are generated in the assembly. And thirdly, which is what we will discuss shortly, is the measurement of the EEG. That is typically performed at the scalp, but it can also be recorded at the cortex, known as a corticogram. And essentially, the EEG is not the collective activity of spiking activity of the neurons, but it measures the much slower membrane fluctuations resulting from synaptic currents as generated in the cortex. So 3 different scales, and in essence different aspects of neuronal behavior are measured with these methods. And clearly, of course, not all methods can be easily applied in human, because of their invasive nature, amongst others. So the EEG is actually a macroscopic measurement where the activity of large neuronal assemblies is recorded, and often many interacting structures are involved as well. For instance, it is known that the thalamus and the cortex have a very intricate relationship, and where this thalamocortical loop is essential for the generation of particular rhythms. So in that sense, the EEG, while it mainly measures the activity from the cortex, also reflects the integrity, amongst others, of the thalamocortical loop. And once more, it's not the single action potentials that is measured with the EEG, but it is, in essence, the membrane fluctuations that result from synaptic currents, mainly in the cortical pyramidal cells. And why is that the case? Well, if we go back to our single neuron, illustrated in this cartoon, this could be an axon or a dendrite, and this is the soma. And there's a single synapse here that, when it's active, will induce a postsynaptic current. And this ionic current that will flow at this position towards the inside of the neuron eventually has to leave the neuron at a particular distance from this inflow side. And as the outflow is typically not completely symmetrical with the inflow, we can define a mean return current of the outflow. And that is typically at another position than the synaptic current. And in this way, we essentially create a small current dipole. So one is either the source of the sink, depending on whether the synaptic current is excitatory or inhibitory, and the mean return current, which is an average of the return current, is typically at a particular position with a distance a from the synaptic current, and thus a very small current dipole is generated. And it is these current dipoles, not a single, but many of them, I will show shortly. It is the activity of all these current dipoles that we eventually measure with the EEG. Now, just to recall, you had this in your physics or bioelectric and magnetic course. The current dipole is composed of a current source and a current sink at a particular distance a, and it results, of course, in currents in the conducting medium that we will assume to be present. And at a particular distance indicated here, we can measure the voltage that results from the current dipole, and the expression for this voltage is given here. You can look in the lecture notes or in the book for more details.